Okay, the floor is yours.
Thank you, Lorenzo. First of all, I apologize for not being able to speak Italian. I feel irrationally embarrassed. I somehow feel I should be able to speak Italian. But I'll try and speak slowly, so if some of you are listening to the interpretation, it'll give a chance for the interpreter to catch up. Secondly, I'd like to express my gratitude to the Fondazione for inviting me here today. I've never been to Alba before, nor indeed this part of Italy, and that was clearly an omission, so I will certainly be coming back. And I've been tremendously excited by the talks I've heard so far today, many of which feed directly into the research I'll present to you today. These are my declarations, none of which affect the content of my talk. You probably don't realize that the previous talk was a masterful expose of 20 years of microbiome research. That was a great job to summarize all of that research so clearly, because there has been a virtual revolution, as Antonio said, in microbiome research. And during that time, I was a normal microbiologist, and I started to study the elderly, because as this cartoon represents, as we heard from Antonio, in the beginning of life, the baby is born without microbes. It acquires a microbiota from its surroundings, and it's accelerated during weaning. And then during adulthood, the microbiome is generally stable, unless there are diseases. And then interestingly, at the other end of life, when people become old, the microbiome starts to decline in lots of different ways. During adulthood, there are many diseases for which microbiome changes are correlated. And I think it's important to be realistic and accept that these are correlations. And the degree of evidence for causation varies from plausible down to pretty bad. So we're still in a phase of trying to really investigate how the microbiome causes disease, if it does. In the elderly, we know from old-fashioned plate and loop microbiology that as people age, their microbiome starts to decline, by which I mean a loss of diversity, a loss of the number of species, a loss of particularly beneficial metabolic activities like degradation of fibers, an increase in the proportional abundance of pathobionts, bacteria which could potentially cause disease. So the backdrop, or the hypothesis for our research when we started this in 2007, was that the microbiome declines with aging. It may be related to food. I come from an agricultural country, and we were particularly interested in the food microbiome link. And we thought, well, if we can find that relationship, perhaps we can stop the decline by dietary interventions. Our first study, which was published 7 years ago now, was a cross-sectional analysis of 500 people in the province of Munster in southwest Ireland. And in these diagrams, each circle is a person. And if two people had the exact same microbiota, those two circles would overlap, which is clearly not the case. And you can see that many people are very far apart, meaning that they have a very dissimilar microbiota. And the point which this slide makes actually is to do with the colors, because in this we have a proportion of subjects who are living in the community, living at home, buying their own food and cooking their own meals. And we also have people in red who are in long-term residential care, and they have a very different microbiota. And we have other smaller cohorts, including people living in long-term care but for less than 6 weeks, people living in the community going to hospitals for short visits, and young controls. So the take-home message of this slide, or this investigation, was that where you live in the community determines your microbiota, which was not something that we expected. And we began to suspect that this was driven by diet, because people living in the community have a high diversity microbiome and a high diversity diet. And when people move from the community into long-term residential care, their diet changes immediately because they have to eat what they are given. But it takes 3 to 6 months for their microbiome to go from a high diversity community type to a low diversity long stay type, which was highly suggestive that the microbiome was driving the change. The other observation was that the difference in the microbiome correlated with differences in lots of health indices. In other words, there seemed to be a link between the difference in the microbiome and frailty, cognition, and levels of inflammation. The weakness in that argument is that these are very different extremes of health already. People in long-term residential care are there for a reason, typically because they're sick and old. So the first thing that we did is we expanded the cohort into people living in the community, and we tried to find people living in their own homes who deliberately ate a bad diet. And this happens for lots of reasons. Sometimes it can be loss of a partner who made all the meals, or sometimes it can just be that these people don't like spending money on food. So all of these people live in the community, and this is the dietary data, and the details are not important. In general, green and brown are good. This is fruit, fibers, vegetables, and stuff in red and yellow is bad. So these people have a healthy diet, these people have a less healthy diet, and these people living in the community have a diet as if they were living in long-term residential care. And when you look at the corresponding microbiome, you see that people who voluntarily consume a long-stay-like diet have a low-diversity diet which is enriched in pathobionts and depleted in bacteria involved in beneficial food transformations such as fiber degradation. Now that's a microbiological observation, and fortunately it also correlates with some of the health measures. So this is an analysis of the functional independence measure, and these two groups on the right-hand side in this panel are the subjects in the community acquiring a long stay-like microbiota, and they have lower values for FIM, which is a measure of frailty. They have lower measures of cognitive function as measured by the mini mental state exam and indeed some other cog tests. They have higher levels of IL-6, which is one of the classical markers for inflammaging, which we'll hear about from Claudia later on today. And there's a trend towards lower values for calf circumference. So this is good. It means that we're not deliberately choosing people at extremes of health, and we can replicate it in the community, but it's still a correlation analysis. It's still not sufficient evidence to prove causation. And to summarize what I've told you so far in this talk, in elderly, we see in many cases a reduction in dietary diversity, which we can measure forensically. We see a concordant reduction in microbiota diversity. We see an increase in inflammation, an increase in frailty. We set about to prove this in preclinical and in human models. The first thing we did was an animal experiment, which I'll describe in detail in a moment, where we did one of these microbiota transplantations, or we humanized germ-free mice and conventional mice that had been treated with antibiotics. And then we gave them different diets to see if we could control their microbiome. And we also did a 6-month fiber intervention with elderly subjects in a variety of different settings. So I'll talk about the mice first. So this is the experimental design, and it's a busy slide, but I'll take you through it, and the message is actually quite simple. We start with healthy community donors, and we're going into mice, and this is what the mouse microbiota looks like. We don't care about those because they're mice. And we treated the mice with antibiotics for 6 weeks to get rid of all their own bacteria. And then this is the microbiota of the human donor. And then over the first 4 weeks, we fed them a good diet, which I've simplified and called low-fat, high-fiber, but actually it emulates the typical properties of the human diet. And what we see is that we can maintain the microbiota of the human donor in the mouse as long as we keep them on the corresponding community diet. Then after 4 weeks, we switched them to a bad diet, which I've summarized as high-fat and low-fiber, and the microbiota changes accordingly. And the microbiota of the mice, given a good microbiome but switched to a bad diet, looks like people in long-term residential care, low-diversity microbiome. Then we did the opposite. We colonized the mice with microbiota from a long-stay donor, or several donors actually. They acquired it quite nicely. We maintained them on the bad diet, and the microbiome stays reasonably constant. When we move them to a good diet, we see a change in the microbiome, but we don't see the restoration of the good microbiome type. Or to summarize this, it's easier to make a good microbiome bad, as shown in the top line, than it is to make a bad microbiome good. And that could be because the taxa which respond to the good diet were not in the human donor to begin with. That's one of the challenges of this kind of research. we weren't just interested in the microbiome, we wanted to see could we replicate any of the differences in human health. We had to do a non-crossover experiment because the crossover would just confuse the readouts. So this is the 4-week experiment where the mice got the community microbiota, or they got the long-stay microbiota, and we're looking at some markers for inflammation here. So the white boxes correspond to the good microbiota, and the dark boxes correspond to the bad. And to be honest, there are no significant differences in these markers for inflammation. We also looked at markers on T lymphocytes, dendritic cells, and macrophages. And in some of these, there's a trend for lower CD4 plus T cells in the mice that got the bad microbiota and higher levels of inflammation. But overall, the differences are quite underwhelming, and there's not good evidence here to support irrevocably the fact that the microbiome is driving inflammation in this animal model. The human part of the experiment was to take human volunteers and to give them prebiotic supplementation for 6 weeks. To change somebody's diet is quite challenging. Old people, like any of us, don't like having our diet suddenly changed on us, so it's easier to supplement or to give them additional stuff. So what we did is we gave them 5 prebiotic fibers which had been published in the literature, which were commercially available, for which we had product liability insurance because we were worried about adverse events. And we had a 2-to-1 ratio of prebiotic to placebo, which was resistant maltodextrin. We ran it for 6 months, which was long and expensive, with a 6-month washout. And we did this in frail subjects because we wanted to improve their microbiome. But we also did interventions in healthy community-dwelling seniors and indeed in a young, healthy group as well. We tried to get an extra 21 grams of fiber per day into these subjects. That probably doesn't sound like a whole lot, but in Western countries, fiber intake can be quite low to begin with, 20 to 40 grams. And if you give somebody of that fiber intake a lot of fiber, they're going to get a lot of bowel upset, and they're going to withdraw from the study. In the next couple of slides, I'll be showing you 3 pairs of panels where you have the young healthy intervention and placebo, elderly community intervention placebo, and then the most interesting group, the elderly long stay intervention and placebo. And this is the microbiome response, the so-called alpha diversity, or one of my favorite metrics, the observed numbers of species. So this is simplified the numbers of bacteria present in those people. And it's lower in the long stay elderly to begin with. And we're looking at the change over time for the 6 months of the intervention and the washout period. And the take-home message is the title of the slide. The prebiotic intervention did not alter the microbiome diversity after 6 months, which was relatively disappointing. But an increasing number of human intervention trials are reporting similar findings. Alpha diversity, a summary statistic of the microbiome, is one of the most difficult measures to change in this kind of way. If you drill down into the detailed microbiome data, you can see changes in individual bacterial taxa. They have a variety of complicated names because they're sequence-based rather than classical taxonomy. So in the top panel, we're looking at bacteria at family level, and lower down, we're looking at genus, which may be more familiar to you. We've flipped the graphs around, so now we have pairs going this way: young healthy intervention placebo, elderly community intervention placebo, and long stay intervention placebo. And this is a heat map where the darker the color, the greater the change in the corresponding microbial taxon. And the first visual impression, which I hope you'll see, is that the greatest amount of change has actually occurred in the long stay intervention group, which is good. Some of the elderly community also changed on the placebo. Maltodextrin is not a perfect placebo. And many of the taxa which changed in the long stay intervention are those which are known in the literature to be responsive to fibers already, things like Clostridium 4, 14A, Eubacterium, Roseburia, etc. So most of these were kind of expected changes in the microbiome, but the fact that something was happening was good. However, when we looked at the inflammatory markers, and we're now looking at the usual 3 pairs of panels, healthy, elderly community, and long stay, There's a trend towards decreased IL-6, which doesn't quite reach significance. It's not significant. There's a temporary trend towards decreased expression of a chemokine CXCL11, which is not significant and not sustained. And there's a temporary increase in CD4 plus T cells. But again, similar to our observations in humanized mice on different diets, none of these changes in innate immune parameters are sufficient to say that a change in the microbiome has a direct causative effect. We kept going because I guess if we were smarter, we would have quit, but we're too stupid to quit. And in contrast to our cousins in England, in Britain rather, we in Ireland, we really like the EU. And we like the EU because it allows you to do large-scale projects. And during this time, we had been lucky enough to collaborate with Claudio Franceschi, who's well known to many of you, and Patrizia Brigidi in Bologna. And they had successfully applied for an EU project called New Age. And New Age was a fantastic project, which first of all did a cross-sectional analysis of over 1,000 subjects in 5 indicated European cities and countries. And then they did an amazingly complex thing, which was to carry out a med diet supplementation of half of those subjects for 1 whole year. The lucky participants had a van called to their house once a week with a box of free food. And it wasn't just a supplementation, because a large amount of their normal diet was being replaced with a Mediterranean diet intervention. And a lot of the fecal samples were extracted in my lab in Cork, and the bioinformatics on the microbiome side has just been completed in my lab in Cork, and we're revising the corresponding paper. The first observation was that the med diet did not alter microbiota alpha diversity in any of the countries. So you can see the data for yourselves. There are some interesting country-specific differences, which I can't show you for reasons of time. France and Italy tend to be more similar to each other and dissimilar to the UK, Poland, and Holland. But in terms of the intervention, there was no whole-scale reprogramming of the microbiome following this 1 year of med diet intervention. Fortunately, we've got some clever people in my bioinformatics group. And what we noticed was that there was a lot of noise in the data, and several of the countries had fairly high levels of adherence to a med diet already at baseline. So if you adjusted for the baseline adherence to a med diet and the baseline microbiota, you could start to see correlations between compliance to the med diet, which was variable, and to changes in the microbiome. And what my talented bioinformatician, Tarini Ghosh, was able to identify were groups of taxa which responded positively to the Mediterranean diet, so-called diet-positive taxa, and groups of bacteria are a group of bacteria which responded negatively, in other words, whose abundance declined as a function of consumption of the Mediterranean diet. And he was able to correlate the changes in abundance of those diet-positive taxa. This is a heat map, so blue is a positive correlation. So those diet-positive taxa correlate positively with levels of constructional praxis, hand grip strength, markers of inflammation. And they correlate negatively with IL-17, gait speed time. So we heard about the 0.8 meters per second walking test this morning. This is actually the time to get from A to B. That correlates negatively because these people can move faster. And the opposite is true. The diet-negative taxa, which are being driven away by the Mediterranean diet, have the inverse correlations. So once we adjusted for compliance and baseline microbiota, we were able to see positive associations with frailty. And the overall onset of frailty proportionally was lower in the med diet intervention group than it was in the placebo. Stefania talked this morning about an ecological approach, and we use an ecological approach to study the microbiome because things like enterotypes, summary statistics, can be a little bit misleading. So this represents the overall microbiome of the entire cohort, and each of these circles is a microbial taxon. And what's interesting is that the diet-positive taxa, the ones which respond positively to the diet, are at the core of these interaction networks, if you like, keystone species. And this is a mathematical proof that the diet-positive taxa are at the center of these interaction networks, and the diet-negative ones are more peripheral. we know from other studies that changing the microbiome with diet is not easy or fast. And we think it's because you have to change bacteria which are interacting with a lot of other bacteria. You're basically trying to rebuild the whole structure of the ecosystem. For the last 5 minutes or so, I want to talk about colon cancer. And we heard from the wonderful expose this morning from Luigi Ferrucci that cancer is one of the second highest causes of morbidity in older people. And in countries like Ireland, where I come from, the rate of colon cancer is somewhere between 5 and 6%, and it's a leading cause of death. And it's interesting that many of the lifestyle factors which correlate with colon cancer risk are known to impact on the microbiome. There are animal models for colon cancer which are suboptimal, but we use them. This is work done by Pat Schloss in the US. And what it shows is that if you treat animals with the mutanogen AOMDSS, you can form tumors in the large intestine. But if you treat the animals with antibiotics, they don't get tumors. So this shows, at least in an animal model, that bacteria are required. And I was skeptical about the role of bacteria in causing colon cancer because it's a long-term 10 or 20-year disease. And I thought it improbable that you could take a tumor biopsy from someone with colon cancer and find bacteria present that were involved in such a long process. Anyway, we did a cross-sectional study, and this is a busy slide, and I just want to draw your attention to this panel here. Again, each of these circles is a person, and what it shows is that there are significant differences between the microbiota of individuals with colon cancer down here and controls, healthy controls on the top. And one of the reviewers asked us to do polyps, which we hadn't included, so we went back and did that. And people with polyps have a mucosal microbiota, which is intermediate between healthy controls and people with actual tumors. I can skip the rest of those. So again, we used an ecological approach, and we divided the kinds of bacteria present into so-called co-abundance groups, or CAGs. And they have complicated bacterial names like Bacteroidetes cluster 1, Firmicutes cluster 1. So there are 6 of these groups which are like the cores in the network I showed you a couple of slides ago. And then we can look at their organization into higher level structures. And we found these structures in German and American published data sets, but they hadn't actually been recognized at the time. And some of these CAGs are more abundant in colon cancer patients, the red ones, and some of them are more abundant in healthy controls. So there's an organizational structure to the microbiome on the mucosa of patients with cancer. So we did a simple experiment, which was we used an animal model which was developed by Irlanda, who's going to talk to you later this afternoon, and colleagues in Israel. It's an orthotopic model. And basically you inject tumor cells and look at the rate of their development. And this is as a function of 2 different microbiome types, and this is a germ-free control. So the mice are going to get cancer anyway. The so-called bad microbiota, which is pathogen-containing, has higher levels of tumors, and then this other microbiota, which is more dominant in healthy people, has lower levels of tumors, except for one mouse. And interestingly, when we go back and look at the microbiome data, we can explain that mouse as a function of the engraftment. It makes complete sense that that mouse has actually more tumors. So we're excited about this, and it resonates to some degree with what you heard from Professor Gasparini on the role of bacteria in modulating the activity of checkpoint inhibitors. The other feature of these microbiome types, when we published this in 2017, we called it the pathogen cluster. But as good microbiologists, we immediately recognized that it was full of oral pathogens, things like Peptostreptococcus, Fusobacterium, Parvomonas, etc. So because they were oral pathogens, we looked at the oral microbiota of a cohort of cancer patients. And this is a PCOA showing that the microbiome in the oral cavity of cancer patients is different from that of polyps and controls. It may not look different to you as a non-expert. The p-value is off scale. They do have a different microbiome. So by this time, we had established a campus company that was interested in artificial intelligence and identifying features for microbiome diagnosis. And we ran some fancy bioinformatics to see, could we use the oral microbiome to detect people with colon cancer? And the answer is we could. Now, colon cancer can be detected by the stool occult blood test, which is pretty poor. There's an immunochemical test, which is much better. So these are receiver-operator curves. And if you've never seen these before, you want the curve to be into this area as much as you can. So this is the microbiome on stool, this is the microbiome in oral, and this is the combination. The bottom line is that a microbiome test on combined oral and fecal detects cancer at about 78% sensitivity and 95% specificity. But for polyps, it's outstandingly sensitive, at least in this highly selected cohort. Now, the fact that we can detect polyps with a non-invasive test is pretty exciting, because for the FIT test to detect polyps, they have to be relatively large, let's say in the range of 1 centimeter. And we don't have to do a colonoscopy, and it could be done cheaply and routinely as part of a routine medical. So we're trying to validate this in a multi-center trial in several different countries at the moment. The other interesting thing about this is that when we look at the microbiome, I've drawn your attention to the fact that we have a pathogen CAG. So these are the people. This is a bit like one of those old microarray slides. These are the people and these are the bacteria. And people who've got bugs which are elevated on colon cancer have loads of this pathogen CAG, and red means lots. And they've got very low levels of organisms which I've called Lachnospiraceae. And you've never heard of Lachnospiraceae perhaps, but they include bacteria which are highly responsive to fibers, fruits and vegetables. So people who get colon cancer have loads of bacteria which are pathobionts, and they lack bacteria which are associated with fiber intake. And that shouldn't surprise you at all, because you know about the links between fiber and cancer. And when we looked at the dietary records of the cancer patients, indeed, they had, in general, lower fiber intake. And I just want to draw your attention to a famous Irish doctor who studied in Dublin in the 30s, and then he went to Africa and discovered Burkitt's lymphoma. But one of his other biggest contributions was to hypothesize about the link between dietary intake and cancer. And we think that part of that link between fiber and cancer could be mediated indirectly through the microbiota, through the kinds of phenomena I've been talking to you about already. So to summarize and conclude what I've said to you today, aging-related species loss is associated with a low-diversity diet. You are what you eat. It correlates with increased measures of frailty independent of where you live. We can replicate diet-responsive taxa in mice, but it's hard to show an effect on health. Long-term prebiotic consumption does have a measurable effect on the microbiota, but borderline effects on health. The long-term med diet in humans is our most exciting finding to date, because it's got measurable effects both on the microbiota and inflammation and frailty. And we propose, and we're not original in suggesting this, that the gut microbiome may be a risk factor for colorectal cancer, reinforcing the known dietary links. I want to conclude by thanking all my colleagues in Cork. We have a microbiome centre with 150 clever people, and these are the people in my lab who do the work. We have a large collaborative network with geriatricians in Cork. The Prebiotic study was sponsored by General Mills, although we didn't actually use any of their products. And I'm hugely grateful to Claudio, Patricia, and New Age colleagues for the opportunity to work on that really amazing project. These are our national funding providers. And last, again, thanks to Fondazione. Thank you very much.